the light-dependent reaction, also known as the light reaction. It is going to involve water, sunlight, and oxygen. Remember, this is taking part in the thylakoid, those kind of green discs that holds the chlorophyll. Sunlight is going to be caught by the chlorophyll, and in this process, the energy harnessed from that is going to split apart water. Oxygen is released as a waste product, and then we have our hydrogens. Now the hydrogens are going to be stripped of their electrons, and the electrons are going to be moving, and it's going to create an electrical current. And that electrical current is going to be harnessed to build energy molecules. Our first energy molecule is NADPH. We have our lower energy form, NAD+. We're going to add those lone hydrogens from the splitting of water, and we're going to form the higher energy molecule, NADPH. And then also from that electrical current, we're going to be able to build ATP. So ADP plus a phosphate to make ATP. Remember, we have that transport protein / enzyme ATP synthase to make that happen. So really, one of the big things from step 1 is we are seeing that light to chemical transformation. Because we start off with sunlight, and then we end up with NADPH and ATP. The whole point of that LDR is to make energy to power step 2. Every reaction needs energy from the start, so our first step is to make energy in order for the second step to even happen. So our second step, we are going to focus on our carbon dioxide becoming glucose. We are in the stroma here, which is kind of this grayish space in the chloroplast. I like to refer to it as like the cytoplasm of the chloroplast. And then we have a couple different names for the light-independent reaction. We could call it the LIR for short. You'll commonly hear it called the Calvin cycle, or sometimes you hear it referred to as the dark reaction. This is simply just to make it different from the light reaction. It doesn't really occur in the dark, it's just that sunlight is not part of this step. So, similar to above, we are still in the chloroplast, specifically the stroma, which is almost like the cytoplasm. The whole purpose of this step is going to be what we call carbon fixation. And in carbon fixation, we have our 6 CO2 molecules, and we essentially are going to make 1 glucose molecule. So 6 1-carbon molecules makes 1 6-carbon molecule. Now, the importance here is our energy molecules from step 1, ATP and NADPH, brought on by step 1, is what is powering that process. And then when this is finally made, what happens then is, to release our chemical energy to power this, we have to cut off that third phosphate. We have to cut off that hydrogen from NADPH. So what ends up happening is we end up having those lower energy forms as products as well, and they are simply going to be sent back to step 1. They're going to be sent back to the thylakoid to continue on that process. So we can always have energy for step 2. So let's kind of break this down. So like I said, step 1, we are using that energy because in step 2, we are making sugar. Stroma being the cytoplasm of the site. So step one, from step one, we have that energy. We are making sugar from CO2 to make our sugar. And then once the sugar is made, we release those lower energy forms back to the thylakoid. So if you want to break step by step, CO2 entering, we break down our energy molecules. We have a series of steps, because remember we have 6 CO2s. So we have a series of steps which are building bonds and kind of putting them all into one thing, which is why it's called a cycle. It's not simply one step. It's a whole bunch of things happening to end up with one molecule in the end. And then once that is formed, we send back your lower energy forms to the thylakoid to get built back up, and the process continues. Light reaction builds the energy, sends the energy to the Calvin cycle. Calvin cycle builds the sugar, sends the lower energy forms back. So just another look at it. The big thing here is CO2, specifically 6 of them, becoming glucose, and glucose is C6H12O6. So everything still kind of taking into account, we kind of break off those H's, and those H's are going to help with the formation of the glucose as well. It's kind of more visual. Big thing, carbon fixation in regards to starting with multiple carbon forms, 6 different carbon compounds, and then we end up with 1 carbon compound, 1 6-carbon compound. All happening in the stroma. There is no chlorophyll in the stroma. It's just where this building is being taken place. Since there's no chlorophyll, there's no sunlight. That's why we call it the dark reaction, in addition to the Calvin cycle. So, which of the following is true regarding the light independent? Very easy to kind of misread that. Independent, the LIR, remember this is the Calvin cycle, or if you want to call it the dark reaction. So which is true? They give off oxygen. That is not true. That is in step 1. Step 1 is the light reaction. They use energy from the light-dependent reaction to make sugar. That is our answer. Let's take a look at the other 2. Letter C. They occur in the thylakoid. No. That is in the first step. This is the second step. They require darkness to occur. No. Even though it's called the dark reaction, it just doesn't need sunlight. So they just refer to it as the dark just to make a difference between the 2 reactions. Not a requirement though. The only requirement is energy and CO2. How many carbon dioxide are required to make 1 glucose? This is where it's important to know our photosynthesis reaction. Simply look at the number. 6 carbon dioxide. There is no number in front of glucose. We assume it to be 1. So we're making 1 glucose from 6 CO2. So kind of big picture thing. First step, we are in the thylakoid. Step 2, we are in the stroma. The big thing with step 1 is sunlight coming in, breaking apart our H2O that releases oxygen, and that energy allows the buildup of our 2 energy molecules. That's going to go to the stroma. In step 2, CO2 comes in. Our energy molecules are broken down through a series of steps. Those CO2s are kind of put together in different forms until finally settling in on sugar, glucose. Once that is finished, our lower energy forms are sent back to the thylakoid to be built back up and start the two-step process over again. So true or false? Photosynthesis will occur in a plant no matter what. Keywords here are no matter what. This is going to be false because we will have some environmental factors, variables that can stop or even slow down photosynthesis. And those factors are going to be basically our reactants. You know, if you don't have water, you can't do photosynthesis. That is a reactant. Light. If you don't have a lot of light, you can't photosynthesize. So we kind of see this with our deciduous trees in wintertime. You know, they lose their leaves because it isn't conducive to maintain them during poor conditions. And then finally, temperature. You know, we do have an enzyme, that enzyme being ATP synthase. If you recall from previous units, enzymes will denature if it is too hot or too cold, and they won't do their job properly. There comes a point of no return where they won't do their job at all. So temperature, big factor with photosynthesis. So water, temperature, light. And we need our ATP synthase to make ATP, which is one of the results of step 1. All right, to summarize, our energy transformation. You know, we are focusing on light to chemical, harnessing sunlight to make it into a usable form. Our first step, our usable form, is ATP and NADPH. And our second step, our usable form, is within the bonds of glucose. And then what we'll learn with respiration is they break down glucose to form more ATP. And then overall, if you want kind of a simplistic way of looking at both reactions. So, let's draw them out. We have our LDR, and this is in the thylakoid. What we have here is light is needed. We also need 6 molecules of water. Water is going to split apart, and we are going to lose 6 molecules of oxygen. In this process, creating a little bit of electrical current, and we are going to build up ATP and NADPH. So that is the chief result. That is being sent to the second step. So this is step 1. This is step 2. The second step is the LIR, light independent reaction. This is in the stroma. What we essentially have happening here is 6 CO2 molecules coming in. We're going to process our energy, cut off that third phosphate, cut off those H's through a whole series of reactions. We end up forming one C6H12O6 molecule. So one glucose molecule formed, and then we end up sending back those lower energy forms of ADP and NADP+, to be built back up. So notice how we have a cycle with our energy just to make sure that we never run out. There's a plan in place to send back the lower forms, build it up in the higher form in the first reaction, send the higher form to the second reaction to help with that complicated carbon fixation process of 6 CO2s becoming 1 glucose, 1 C6H12O6. All right, that marks the end of photosynthesis. Thanks for watching. Let me know if you have any questions.